Hello everyone, welcome to BiotechAcademy's Immunology 101 series. In this video, we are going to talk about the innate immune system. Let's get started. The innate immune system is our body's first line of defense against infection. It responds rapidly and non-specifically to any pathogen, acting as a general defense mechanism. So this is important. Whenever you hear innate immune system, you should know that it's rapid and non-specific. There are different types of barriers. For example, physical barriers or chemical barriers. The most prominent barrier is the skin. In addition, there are mucous membranes, lining our respiratory, digestive tracts, and actions like coughing, sneezing, or saliva and urine physically expel invaders. Chemical defense, on the other hand, reinforce these barriers. For example, the skin produces sweat and oils, inhibiting bacterial growth. Our stomach secretes stomach acid. Enzymes like lysozyme in tears and saliva can break down bacterial cell walls. Even our normal microbiota, so the good bacteria living in our skin and gut, serve as a barrier by outcompeting harmful microbes. Now I'd like to talk about the major innate immune cells. Once a pathogen penetrates the initial barriers, the innate immune system deploys various immune cells to quickly attack and contain the threat. These cells are often called white blood cells, but we will use the term leukocytes. We are going to talk about macrophages, neutrophils, dendritic cells, natural killer cells, mast cells, basophils, and eosinophils. Let's start with the macrophages. So the name macrophage literally means big eater. When a macrophage encounters a microbe, it engulfs, aka swallows, and digests it. So as you can remember, a phagocytic cell has the capability to engulf a microbe and then destroy it into little pieces. So instead of saying big eater, we're going to use the term phagocytic cell. They attack dead cells, and this one is important. Release cytokines. What are cytokines though? For now, you should know that cytokines are little signaling proteins which recruit other immune cells. Neutrophils are the most abundant type of leukocytes in circulation. So what we should know is they are often the first responders to an infection site. Then you should also know that they're phagocytic, but they kill bacteria. Neutrophils are short-lived foot soldiers. They swarm into battle, release toxic substances, for example enzymes and reactive oxygen species, to destroy pathogens. The accumulation of dead neutrophils and debris can form pus, the yellowish fluids seen in infected wounds. Despite their short life, neutrophils are crucial for rapidly containing bacterial infections. Next one would be the dendritic cells. These cells act as the messengers between the innate and adaptive immune systems. Dendritic cells reside in tissues and also perform phagocytosis, capturing bacteria and viruses and digesting them. So dendritic cells start out in peripheral tissue. So this is tissue located away from the central body, and these are not directly involved in the primary function or an organ system. After engulfing a pathogen, right here, dendritic cells travel to lymph nodes and present pieces of the pathogen, so to say antigens, to adaptive immune cells, in this case T cells, to initiate a specific adaptive response. Next, we have natural killer cells. So NK cells, natural killer cells, are a type of innate lymphocyte, a white blood cell related to T and B cells, but part of the innate immune system. They specialize in recognizing and destroying abnormal host cells. For example, cells that have been infected by a virus or turned cancerous. The reason for that is that cells infected by viruses, or the ones that turn cancerous, have a tendency to increase their amount of receptors, and the natural killer cells are able to detect these. If a cell is missing certain self-markers or has other distress signals on its surface, the NK cell will release toxic granules called perforin and granzymes that induce the infected cell to die, basically a form of cell suicide called apoptosis, cell death. Lastly, we will talk about mast cells, basophils, and eosinophils. These are other innate cells, often called granulocytes, that play supporting roles, especially in fighting larger parasites and in allergic responses. So we've learned that mast cells release histamine. Then there are basophils. They are less about ingesting pathogens and more about bombarding pathogens or signaling other responses. In summary of cells, rapid identification is key. Our phagocytic cells are macrophages, neutrophils, and dendritic cells. Then we have other cells, which are NK cells, mast cells, etc., like granulocytes. So here we have a question. How do these innate cells know when to spring into action? Pathogen recognition via PRRs and PAMPs. Let me explain. So one of the remarkable features of the innate immune system is that it can instantly detect that a microbe is present even if the body has never seen that microbe before. It does this by recognizing certain molecular patterns that are common to many pathogens. Innate immune cells have built-in sensors called pattern recognition receptors, shortly PRR, on their surface or inside the cell. So let's give some examples for molecular patterns. Lipopolysaccharides, shortly LPS. Flagellin from bacteria can also be an example. So PRRs are designed to bind to pathogen-associated molecular patterns. Let's say we have a bacteria, and those bacteria have PAMPs, pathogen-associated molecular patterns. Molecules that are typically found on bacteria, viruses, or other pathogens, but not on our own human cells. For example, bacterial cell walls often contain a molecule called peptidoglycan. And gram-negative bacteria have lipopolysaccharides, LPS, on their outer membrane. Our innate immune receptors recognize these. For instance, toll-like receptors, TLRs, are a well-known family of PRRs. For example, there is something called TLR4, which recognizes LPS from bacteria, and TLR5 recognizes flagellin. As soon as PRR binds its target pump, it sends an internal signal to the immune cell, alert, a pathogen is here. So the recognition of a pathogen causes the activation of macrophages and dendritic cells and start producing cytokines. But now the question arises, what are cytokines? Let's answer that question. Cytokines and chemokines. When an innate immune cell recognizes a pathogen via the PRRs, it quickly releases chemical messenger proteins called cytokines. Cytokines are signaling molecules that allow immune cells to communicate with one another. Cytokines act on other cells by binding to receptors and telling those cells how to behave. For example, to become active, to move to a location. A special subgroup of cytokines called chemokines has the specific job of attracting immune cells to the site of infection. The term chemokine comes from chemotaxis, which means movement directed by a chemical gradient, like a scent that other immune cells follow to find the exact location of the problem. Some key actions of cytokines in the innate response include promoting inflammation. So certain cytokines, for example, tumor necrosis factor or interleukin-1, make blood vessels leaky and recruit immune cells, causing the classic signs of inflammation. Another thing cytokines can cause is fever. Cytokines like interleukin-1 or interleukin-6 act on the brain to increase body temperature, resulting in fever. Another thing is recruiting and activating cells. Cytokines can call in various cells and ramp up their killing ability. For example, interferons, which are a type of cytokine, are released by virus-infected cells to warn neighboring cells and help render them resistant to the virus. I'd like to talk about cytokines' ability to contain infection. Some cytokines even help form clots in small blood vessels at the infection site. Others instruct liver cells to produce acute phase proteins that aid immunity. Now, here we are talking about the complement system. The complement system is a collection of proteins in the blood and tissues that complement the cellular innate immune response. Think of complement as a series of falling dominoes or a chain reaction. Once the first protein is activated, it activates the next, and then the next, and then the next, and so on. This cascade results in a powerful antimicrobial outcome. Major functions of the complement system include opsonization, chemotaxis, and direct killing. So let's explain each one of them in greater detail. Let's explain opsonization, the tagging of pathogens. Some complement proteins stick to the surface of invading microbes, marking them as targets for phagocytes, as if complement paints a big red X on the pathogen. Phagocytes have receptors that recognize these complement tags, making it easier for them to bind to and engulf the pathogen. Here in chemotaxis, other complement fragments diffuse away and act as chemotactic signals, attracting immune cells to the site of infection. This ensures more phagocytes and other defenders to arrive quickly. Then we have direct killing, the so-to-say membrane attack complex. The final group of complement proteins can insert into the membrane of a microbe and assemble into a pore-like hole, known as the membrane attack complex. MAC. This punctures the microbe's cell membrane, leading to the leakage of contents and death of the pathogen. The complement system can be activated in a few different ways. One way would be the classical pathway. The other one would be the mannose binding lectin pathway. And the last one, the alternative pathway. We will discuss them in a separate video. Let's talk a little bit about inflammation. Inflammation is essentially the body's way of raising the alarm and marshalling defenses to a local area of infection or injury. It has several signs. The first one being redness and heat. The other one being swelling, maybe even pain. Let's break down how these occur. When innate immune cells, like macrophages and mast cells, detect pathogens or tissue damage, they release inflammatory mediators, including cytokines. Yes. For example, TNF. TNF was tumor necrosis factor, or interleukins. For example, interleukin 1. These substances cause nearby blood vessels to widen and the vessel walls to become leaky, aka more permeable vessel walls. As a result, more blood rushes to the area, hence the redness and warmth. The leaky vessels allow fluid, plasma proteins, and white blood cells to exit the bloodstream and enter the affected tissue, leading to swelling, which is called edema. So let's see what happens inside of our body. So inside the inflamed tissue, neutrophils and other phagocytes begin gobbling up bacteria and debris. Complement proteins are activated and assist in destroying pathogens. The increased blood flow brings more oxygen and nutrients to support the immune cells in the bowel. Pus, if it forms, is an accumulation of dead cells, mostly neutrophils and microbes, a sign that the body is fighting the infection. However, too much inflammation can be damaging to the body's own tissues. For example, if an infection spreads through the bloodstream and it triggers inflammation everywhere, it can lead to a dangerous condition. For example, septic shock due to an overwhelming cytokine release. So lastly, I'd like to talk about the innate immunity versus adaptive immunity. So first of all, I'd like to point out the speed of response. So the innate immunity responds immediately, versus the adaptive immunity needs its time. The innate immunity is non-specific, whereas the adaptive immunity is specific. So the innate immunity has no lasting memory, versus here, actually memory cells can form. Lastly, I'd like to talk about the development. Innate defenses are hardwired in our germline DNA and are ready to go from birth. Adaptive immunity is developed over time. As we get exposed to pathogens, each person's adaptive immune repertoire is unique based on what their body has encountered or what they've been vaccinated against. So this was it for today's video. Please don't forget to like and subscribe to Biotech Academy. I really put a lot of time and effort into these videos, and I'll see you in our next video.